All right, good afternoon, everybody. It's Thursday, and as I mentioned, I'm going to be recording the first respiratory lecture. We will be having another one on Tuesday. I'm just not sure of the content yet, so I will be updating that and then posting that on Canvas. So anyway, like I mentioned, we're also going to have a review for the Thursday exam on Tuesday, and I'll send that out to you as well. All right, so let's go ahead and start with the respiratory lecture. Oops, let's see, what am I doing here? And there we go. All right, so a nice follow-up to the cardiovascular system, or I should say a logical follow-up, is the respiratory system. Because of course we talked about the cardiovascular system as far as delivering oxygen, removing CO2. It's a transport mechanism, right? But we've got to somehow bring in oxygen and get rid of CO2 in reference to the outside world. So we're going to talk about this very strong correlation between those two systems. This particular lecture is going to be more focused on anatomy, structures, some functional features of different part of the respiratory tract. And then on Tuesday, we'll focus more on some of the mechanics, the mechanism of breathing. All right, so every system, of course, has a function or many functions. The primary function of the respiratory system, of course, is gas exchange between the atmosphere and the blood. And of course, if you think of the gases, you think of oxygen traveling from the outside world or the atmosphere, gets into the blood, circulates to the body, gets dumped off on the tissues, and by the same token, CO2 from the tissues gets transported to the blood and then goes out to the atmosphere. So they're opposing pathways. Based on the relationship between oxygen and CO2, we'll soon learn that that actually dictates pH. You guys remember from your physiology lab when you were looking at buffers and you looked at carbonate buffer, carbonic acid. And of course, carbonic acid is formed from carbon dioxide. So one thing we know, of course, is that the more CO2 there is, the more acidity there is in the blood. And so we obviously have to regulate the pH of the body, maintaining it into a typical 7.2, 7.4. And that's done by how we breathe. Thirdly, and this is primarily due to the structures of the respiratory system, while we're breathing oxygen in, we're also bringing in the rest of the air. And the air, of course, even though it's not too bad in San Diego compared to LA where I grew up, there's particles, stuff that we don't want. And fortunately, we have structures in our respiratory tract that can actually filter and remove substances that we don't need or don't want. Lastly, and I'm just mentioning this in passing only, is structures of the respiratory system, such as the larynx, play a role in vocalization. Of course, the larynx is the voice box, as well as being a conduit for air. All right, just some terminology to get things started. We will use the term ventilation and breathing and respiration many times. What do they exactly mean? Well, ventilation is just a general term to describe the process of breathing. What is breathing? Breathing is the cycle of inspiration, bringing air in, and expiration, or removing air out of the lungs. Now the gas, there's two different drop-off points for the gas, or two different areas where it moves. One, of course, if we think of oxygen coming from the outside and being delivered to the lungs and then eventually into the blood. That's what we call external respiration. And then from the blood, that oxygen then is transported to the tissues. That would be internal respiration. And by the same token, carbon dioxide, the internal respiration is moving CO2 from the tissues back to the blood. And the external respiration is moving it from the blood out of the lungs. All right, so taking a closer look at internal and external respiration, you can see the two different parts of it. Here, pretend this is the outside world, and here is oxygen being brought in. These are structures called alveoli, which we'll soon learn about. From the alveoli, if everything is working properly, oxygen is delivered into the pulmonary circulation. This would be the pulmonary veins, which would then bring the blood back to the left atria, left ventricle, of course, then out into circulation, where it's transported and eventually dropped off at the tissues. So this down here, the drop off at the tissues is internal respiration. Movement from the outside into the blood is external. And then, of course, we have CO2 that builds up just from normal metabolism in organs, delivered back into the venous blood, back to the right side of the heart, where it eventually goes out through the pulmonary artery, and then we breathe it out. All right. Just some other features of it to provide kind of a numerical aspect of breathing, because much like the blood, breathing is determined by pressures as well. So as we learn, external respiration involves movement, and really more correctly, the diffusion of oxygen from the lungs, these are the lungs, into the blood, and the diffusion of CO2 from the blood back to the lungs. Now, we know from diffusion is the movement of something from a high concentration to a low concentration, or from a high pressure to a low pressure. Well, let's take a look. Here in the lungs, the pressure of oxygen is about, where's my pointer? Is about 100, is 100. It diffuses into the pulmonary circuit, where right about here, these are the pulmonary capillaries, it's about 40. So 100 to 40, very easy to diffuse. Once, of course, the oxygen gets into the blood, the oxygen is now at 100, and that's where it stays at until it starts dropping oxygen off at the tissues. What about CO2? Well, the blood level of CO2 is roughly around 45 millimeters of mercury. And what happens is when it reaches the right around the lungs, the blood vessels of the lungs, compared to in the lungs themselves, this is the blood vessel, these are the lungs, the CO2 is 40. So 45 to 40, not that dramatic of a difference, but enough of a change that's going to allow for diffusion. So that's external respiration, right? Going from the lungs, between the lungs and the blood. Internal respiration, we're still going to see the diffusion, but it's obviously through different area. Here we have diffusion of oxygen, which is at 100. Now it's in the blood into the tissues. The tissues is at 40. So from 100 to 40. CO2, which was at 45 at the tissues, CO2, which was at 45, originally in the blood, if you go back up here, it's at 40. So from 45, it goes to 40, and then eventually, obviously, it becomes 45 as it equilibrates. All right, so this is kind of putting everything together. Numbers are a little bit different, right? Instead of 45, we have 46, but everything else is basically the same. Oxygen from 100 goes into the blood when it's 40, brings it up to 100, then it gets dropped off. CO2 is about 46, 45 in the tissues, gets dropped off into the blood when it's 40, brings it up to 46, goes to the lungs where it's at 40, and it just diffuses out. All right. So a lot of what the respiratory system does is, no surprise, it's dictated by the anatomy, the structures. What is it about the structures of the lungs that enable it to do a lot of these things? Well, if you take a look at the entire respiratory system, not just the lungs, the entire respiratory system begins at the nose, right? That's where we breathe in, or that's where we're supposed to breathe in. We really shouldn't be breathing in through our mouth. That's part of our digestive system, and we don't have the filtering capabilities there. We breathe air in through the lungs, or the nose. It then travels, as you can see, it's boxed in here, through what's called the internal nares. This is like a passageway into the pharynx, So nose, internal nares into the pharynx, which is this common cavity behind the mouth, behind the nose, and then we have the larynx. Just so kind of follow the path. From the larynx, there's that epiglottis. Some of you guys might remember when we were talking about elastic cartilage in anatomy. From the larynx, the air then flows into the trachea, then branches out into the bronchi, and eventually into these much smaller structures called bronchioles, and then finally into the alveoli. And it's the alveoli where true gas exchange is going to occur, where the oxygen is going to be diffusing into the blood, and where CO2 is going to be diffusing from the blood back into the alveoli. All right. So we've already spoken about surface areas, right? Surface area in terms of blood flow. Remember, increased surface area in the capillaries decreases the velocity. Well, one other aspect of surface area is that the greater the surface area optimizes the function. So with the capillaries, even though the blood is very slow, that large surface area allows for a lot of oxygen delivery, right? In the case of the airways, we have a lot of branching, not of vessels, but of the bronchial tubes or the airways. We have the larynx, the trachea, which branches off into what we call the primary bronchi. And these branch several more times into the secondary bronchi, into the tertiary bronchi. You don't see it here in the bronchioles, ultimately branching 22 times roughly, ultimately into the alveoli. So this branching, much like we saw from arteries to arterioles to the 40 billion capillaries, right, greatly increases the surface area, which in this case will maximize the ability for gas exchange between the lungs and the blood. All right. So just to give you an idea of the surface area or the cross-sectional area, we can take a look at the different parts of the respiratory tract and also the numbers of these different parts. If you take a look, first of all, we have the trachea. Obviously, we have the nose and other things and the larynx, but let's start with the trachea. We have one trachea, right? It has roughly a cross-sectional area of 2.5 square centimeters. We then branch into the primary bronchi, of which there are two, right? And those bronchi divide, or I should say don't divide, they actually branch several times, ultimately going from 2 bronchi to 4 bronchi to 10,000 bronchi, which greatly increases the surface area as well. Ultimately, those bronchi then branch further into the bronchioles, of which there are anywhere from 1 to 23. I've never seen one, but multiple branches, which leads to an increase in surface area of about 5,000, and there's about 80 million bronchioles. Ultimately, this branches out further into the alveoli. There's 24 branches of the alveoli, which results in roughly about 300 million alveoli. That's per lung, which leads to a surface area of greater than 1 million. So a lot of branching, greatly increasing the surface area, which is ideal because by the time we get to here, this is when we want to do the gas exchange. Notice also on this side, I speak about what's called the conducting zone. Hold on a second here. Never mind, I want to make sure I was recording. Nope. We have the conducting zone and the exchange surface, or the exchange zone, or sometimes called the respiratory zone. The conducting zone refers to the part of the respiratory tract where we're just transporting the air, maybe filtering it, cleaning it up, but we're not moving it into the capillaries. It's only right around here where we have some of the smaller bronchioles and the alveoli where that air that's been filtered is now moving into the blood. All right. So this is just kind of a side-by-side comparison. You're going to hear me talk a lot about surface area, how with lungs we have roughly 600 million alveoli, large surface area. Circulatory system, we have large number, large volume, large surface area of capillaries. And later we'll look at the intestine, large surface area. All these large increases, large surface areas are very important in contributing to the function. With the circulatory system, like I mentioned, it just certainly slows the blood down, but it also maximizes the movement of blood into the tissues. With the lungs, in this case, we're maximizing the gas exchange between the lungs and the blood. And with the intestine, this is going to maximize the amount of absorption of nutrients. All right. All right, so a little bit of anatomy. Where are the lungs located? They're located in the thoracic cavity. Thoracic cavity, of course, is one of the ventral cavities that's separated from the abdominal pelvic cavity by this important muscle called the diaphragm. We'll learn more about that later. This pink structure is the lungs, but notice that there are layers over it, on top of it. These are what are called the pleural membranes, and there are two of them. Once again, in anatomy, we've spoken about serous membranes, right? Serous membranes that wrap the heart, like the pericardium. Serous membranes that wrap some of the organs in the digestive tract, the peritoneum. And here you've got the pleural membranes, of which wrap the lungs. And there's two of them. The innermost is called the visceral pleura. The outer one is called the parietal pleura. And in between them, we have a fluid-filled cavity called the pleural cavity, which plays a very important role in reducing friction that can occur during breathing between the different membranes. Anytime you've got a moving structure, just like even moving parts of an engine, there's friction. So with the engine, we've got oil, right, to reduce the friction. Here we've got this lubricating pleural fluid that reduces friction. And much like with the heart, we've got the fluid surrounding the myocardium that helps to reduce friction as the heart beats. All right. So as I mentioned, we've got two different parts of the respiratory tract. We've got what's called the conducting zone and the exchange zone, or the respiratory zone. The conducting zone starts with the nose and continues down through the trachea, bronchi, all the way to what we call the terminal bronchioles. In contrast, the exchange zone, or the respiratory zone, starts with the respiratory bronchioles, which are branches off the terminal, and then eventually ending up with these grape-like clusters called the alveoli. And this is where the gas exchange is going to occur. All right, so what is the purpose of the conducting zone? Well, first of all, like I mentioned, it starts from the nose, goes all the way down to the terminal bronchioles. Its function is really air conditioning, basically cleaning the air and preparing it for gas exchange. So what does that air conditioning involve? It involves warming the air and humidifying it, right? The air outside is not at the same humidity as it is inside. And if we bring in dry air and don't humidify it, that could cause damage to our tissues. So we need to warm it, moisten it, and at the same time, filter out any dust or any particles that we don't need. This is where some ciliated epithelium comes in handy, and we'll talk about that as well. You guys are also familiar with that from anatomy. So a lot of anatomical, this is Throwback Thursday, I guess. All right, so taking a look, this is a perfect example of what we call the respiratory epithelium, or the lining of the respiratory tract. And the size of the cells will vary somewhat, but typically we have these ciliated cells interspersed with what are called goblet cells. And the way much of this cleaning works is these goblet cells will squirt out mucus. So this is the lumen. This is where air is flowing. Will squirt out mucus, which traps the junk, and then the cilia will beat, and they'll basically will sweep up the mucus, eventually will be expectorated, and then, not pleasant to hear, then we swallow it, go through our digestive tract, and enzymes break down the bad stuff, right? So it's a way of expectorating. People who have cystic fibrosis, which is a topic we'll cover on Tuesday, unfortunately, they have a very, very thick mucus. And as a result, those cilia are unable to move it. And so as a result, a lot of the nasty stuff, dust, even bacteria, get trapped. And because of that, people who have CF are very vulnerable to infections. All right. So going along with the conducting zone, let's take a look at the kind of the general histology of the trachea and the primary bronchi. If we were to slice through the trachea, one of the things that you would notice is essentially the epithelium. If you remember the lining epithelium of any cavity, right? In the case of the trachea, it's pseudostratified ciliated columnar, otherwise known as PSCC. So it's got the cilia. As we transition into the bronchi, primary bronchi, the cilia, instead of being pseudostratified, becomes more simple columnar and can also be ciliated as well. One feature that's common to both the trachea and the bronchi is we have these rings, right? You can't see you can see the ring here. You can't tell that it doesn't go all the way around, but it makes it what's called a C ring for that purpose. This is hyaline cartilage that helps to keep the airway open, keeps the airway kind of stiff, because otherwise the airway would collapse in between breaths. And there's a neat little muscle right here. One of the reasons why we don't want the cartilage going all the way around is then it would push on the esophagus, which is part of the digestive tract. All right. Moving from the bronchi to the bronchioles, we see some other changes. And this is kind of comparable to when we're talking about blood vessels. Remember blood vessels, we spoke about 3 layers, arteries, and then 2 layers with arterioles and venules, and then 1 layer with capillary. Here we're seeing this transition. Why is this transition occurring? Much like what we saw with blood vessels, there's going to be a change in function. With arteries, remember the size of them, the thickness is elasticity, withstand the blood pressure. As we get down to the arterioles, we're more concerned with regulating blood pressure due to the high percentage of muscle and also reducing the blood flow, which was fast before that. And then the capillaries, very thin for gas exchange. Here, the bronchioles are certainly bronchi are getting smaller as we reach the bronchioles. These are the bronchioles, these are the bronchi. Notice with the bronchi, even though the bronchi, we see this C rings like we saw with the trachea, as we start transitioning to the cartilage becomes more like patch or plate-like. See this? And then eventually the cartilage disappears and we have the bronchioles, the terminal as well as the respiratory bronchioles. So what are some of the changes? Going from the trachea and bronchi to the bronchioles, there's first of all, the epithelium is gone from PSCC to columnar, now to cuboidal. We lose the cartilage, and there's an increase in the proportion of smooth muscle. The bronchioles are kind of like the arterioles because these help to regulate airflow. And people who have asthma are very familiar with this because during an asthma attack, these muscles constrict, which make it difficult to breathe. And finally, of course, it's a smaller diameter. Finally, we're at the point where we're at moving from the bronchioles, oops, sorry about that, from the bronchioles. And basically the terminal bronchioles is the end of what we call the conducting zone. Now we're moving into the respiratory bronchioles and the alveoli. We're in the respiratory zone. The epithelium now changes again. It goes from cuboidal, simple cuboidal, to simple squamous. And this is the site of gas exchange, much like the capillaries with their thin epithelium is also the site of gas exchange. Gas exchange, as you know, occurs by diffusion, right? Going from an oxygen content of 100 and it flows into where it's 40, right? In the blood, but then it increases it up to 100. The importance of this respiratory zone, of course, is the fact what allows respiration to occur so efficiently is not only because the cells are very thin, but because they're surrounded by capillaries, by blood vessels. So the oxygen does not have to flow very far from the alveoli into the capillary. It's a very short diffusion distance. All right. So now we're at the alveoli. So what do we know about the alveoli? Well, the alveoli are these like little grape sacs that are kind of part of a cluster, but each individual one is called an alveoli. The whole thing is called a sac. Well, in each of these sacs, this is of course where gas exchange is going to occur. And here we've got a cross section, say we're looking right in this area. This is the alveoli, so air is coming in here. Here's the alveoli. Here's the capillary, which is this right here. The oxygen is going to flow by diffusion into the capillary. CO2 is going to flow by diffusion from the capillary back to the alveoli and then back out, right? We breathe it out. This is a very thin surface. Like I mentioned, you want a thin surface for rapid diffusion. And that thin surface consists really of three parts. We have the alveolar epithelium, or the simple squamous cell. We have the capillary epithelium, which is sometimes called endothelium, but two very thin cells. And then in between that, we have this kind of a glue, which is called the basolamina. So as long as that stays thin, which it usually does, we have rapid diffusion. People who have conditions such as pulmonary fibrosis or other types of conditions, this actually will get scarred, and this will make it a much thicker layer, which will make breathing more difficult. This alveolar cell that we're talking about is sometimes called a type 1 alveolar cell. And of course, this is what the oxygen has to flow across to get into the capillary. But we also have a couple other cells. We have a type 2 cell, which is known as a surfactant cell, producing cell. Some books also call it a Clara cell, C-L-A-R-A. This produces surfactant, which is like a detergent, which reduces the surface tension, which I'll explain that in the lungs, which as a result prevents the lungs from collapsing. Finally, we have another interesting cell called an alveolar macrophage, or just a macrophage. And this is kind of like the macrophages that we see in the brain, the microglia. These are little scavenger cells that kind of go around doing housekeeping. Even though the air is pretty clean, there could be other stuff that builds up in here. So the macrophages are another type of cell. So we have three types of cells. This first cell is important, forming part of what we call the respiratory membrane, along with the capillary. We also have the surfactant cell, of course, and then the macrophage. All right, so let's talk now about the mechanics of breathing. So we know about the anatomy. What is it about the anatomy? What is the mechanics of causing us to breathe? It's unconscious, right? We don't have to think about it. Well, there's several aspects of it, obviously starting with the brain, which we'll get to later. But one of the most important things that governs breathing is, again, a pressure-volume relationship. At a fixed temperature, there's a law, another law. We've spoken about Puiseuil's law and Fick's law, remember those? This is Boyle's law. basically discussing the relationship between pressure and volume, looking in two different closed chambers, basically saying P1 times N1, or the pressure times the volume in one chamber, is going to be equal to the pressure times the volume in another chamber. These are kind of continuous chambers. If we translate that, we can basically simplify it and say that pressure is inversely related to volume, which we already knew when we were talking about the respiratory pump moving air up to the heart. So air moves from high pressure to low pressure. This is what the alveoli look like in different conditions. When you're breathing in, the alveoli increase in size, as you might expect. When you breathe out, they decrease in size, as you would also expect. All right. So let's take a look more at this whole pressure-volume relationship and how it occurs in different phases of breathing. Basically, there's three parts to the breathing cycle, right? We have at rest, which would be the brief period between inhaling or inspiring and exhaling or expiring. If we take a look at the pressures, two main pressures we're looking at. We're looking at the outside pressure, the atmospheric pressure, which is at 760 millimeters of mercury. And then if we take a look at the pressure inside the lungs, or the intrapulmonary pressure, at rest, that's the same. We'll talk a bit about intrapleural pressure later, but these two pressures are equal, so there's no net movement of air. During inspiration, what happens is atmospheric pressure doesn't change, right? We have no control over it. Well, it does change, obviously, with weather conditions, but very minuscule. So it basically stays around 760. During inspiration, look what happens to the intrapulmonary pressure. It went from 760 to 757. Doesn't seem like much, but it's enough to draw air in. High pressure to low pressure. When we breathe out, notice what happens again. The pressure here is, the pressure in the lungs is now 763. It's more than the outside, so it's going to move air out. Okay, so that's all well and good. We have this difference in pressures, but and it's a difference of 3. If we take a look, for example, during inspiration, the inside, the intrapulmonary pressure is -3 compared to the outside, 757 versus 760. And during expiration, or just prior to expiration, the inside is 763 compared to 760, so that's a +3. We're going to kind of, for now, just ignore these lower two. All right. So taking a look at this, and then we'll get to the explanation. Like, you might wonder, well, why is the pressure changing? What's going on? Okay. Well, before we get to that, I want to give you a neat example. And there's a really good online video. I think I also put the video on the Canvas page you can take a look at. It's called the Marshmallow Man video. I didn't put the video in here, but just wanted to illustrate that if you take this is actually a flask that has basically, what was I going to say? Yeah, so this is a flask with a marshmallow in it. This is the same flask with the same marshmallow, but we've applied a vacuum. All right. So when we applied a vacuum, what did that do? Well, when we applied a vacuum, we reduced the pressure. When we reduced the pressure inside, what happens to the marshmallow? It got bigger. This is analogous to the pressure inside, the lung pressure decreasing, which is going to cause this to increase. Okay. The video really shows it much better. So decrease pressure, increase volume. And the video is pretty cool. They actually show this marshmallow man exploding, right? If you ever saw Ghostbusters, or the first one, the Stay Puft Marshmallow Man. So anyway, this is all Boyle's law. Pressure of a gas inversely related proportional to the volume. Increase in lung volume decreases decreases intrapulmonary pressure, causing inspiration. So this occurs during when you're breathing in. The lung volume increases first, which decreases the pulmonary pressure, causing us to breathe in. When you're breathing out, first, there's a decrease in volume, which leads to an increase in pressure, causing expiration. So notice how I emphasize these two things occurring first. So the question is, what's causing the lung volume to change? What's causing the lung volume to decrease or increase? Okay. Well, one of the things we do know, it's related to muscle contractions. And we'll talk about those later. Okay. Contractions of the diaphragm, contractions of the external intercostal muscles, which affect the size of the thoracic cavity. When those two muscles contract, diaphragm and external intercostals, that causes the volume to increase, which decreases pressure. When those muscles relax or even relax even more so, what happens? That causes the volume to decrease, which causes air to be pushed out. All right. So one of the things we know about for the lungs to work properly, in addition to having obviously everything intact, the trachea, the bronchi, everything delivering oxygen to the lungs, that's well and good, but there's got to be certain properties of the lungs that enable it to inflate, much like we saw right there. Oops, why is that doing that? Right here, right? So first of all, we need three main properties for the lung to properly inflate or to ventilate. First of all, we need the lungs to be compliant. That has to do with the ease of inflating, and we'll talk about what causes that shortly. Second of all, the lung has to be elastic. Once it inflates, it has to be able to go back to its original size to be ready to inflate again. And thirdly, there's a certain amount of surface tension that's in the lungs that needs to be minimized. And that's where we'll see the surfactant comes into play. All right, so what do we know about compliance? Compliance is the ability to stretch under pressure. And compliance is greatly reduced by factors that cause resistance to distension or stretching. For example, if someone has what's called black lung or pulmonary fibrosis, there's a lot of scarring. When there's scarring, the elastic tissue has broken down and the lung cannot inflate as easily. That's what we would see right here. When everything is working normal, the lungs inflate very easily, right? The elasticity allows it to inflate and then also deflate very well. This is a pathological condition right here. So if you have decreased compliance, it requires more force to breathe. And this is pretty much can be related to what we call restrictive or fibrotic lung disease. This is due to people who are exposed to, again, either coal or even asbestos. Obviously, asbestos causes other things, certain cancers, mesothelioma, but it also can directly cause this fibrosis on the lung. And also the failure, one thing that can also affect compliance is the inadequate surfactant production. So we'll take a look at that. Without surfactant, the lungs also don't inflate as well. What about elasticity? Elasticity is allowing the lungs to return to their original size after stretch. So without elastic tissue, or if it's broken down, the lungs will not be able to inflate or deflate very well. What does that translate into? We can't bring in as much air as we would like, oxygen, and we can't breathe out as much CO2 as we need to. So these dark fibers are all elastic fibers that are found. This is alveoli right here. These are all these elastic fibers. All right. What about surface tension? So this is a loaded topic. We'll get into it more when we're talking about cystic fibrosis, as well, more details about it. But as far as the basics, surface tension is something that will have a detrimental effect on the lungs. It promotes collapse of the alveoli and increases the resistance to distension, which means it's more difficult to inflate the lungs. All right, so what is this surface tension? Well, you guys are all familiar with surface tension. We think of hydrogen bonding water molecules. If you've ever done a belly flop in a swimming pool, it hurts, right? That's because the water molecules are forming a bond with each other, forming kind of like a sheet. Same thing is true if you have like a water in the back of your yard or a pond, and you see these little water skippers or mosquitoes walking across the water. They're able to do that because of the surface tension. If you wanted to, this would be, I don't want to get in trouble from animal rights or whatever. If you, in this little pond, if you take a little bit of detergent, that breaks the surface tension and the mosquito will just be pulled under. But too bad this is recorded. Anyway, so basically surface tension is created by hydrogen bonding of water molecules. So where does water come from? Well, water is floating all around many of our tissues, right? If you go back here, we have water, fluid. And this is due to the fact that much like any organ, there's a constant influx and efflux of ions, right? Typically, lungs secrete and absorb ions in water. There's this constant interplay. We're not going to talk really about CF right now. This is where this whole situation is impaired. Basically, it amounts to the lungs are not secreting chloride ions in the right direction. And as a result, there's no water that goes out. So the mucus gets very sticky. Remember, I mentioned that there's mucus that line the tubes. They also line the lungs. So, but we'll get into that later. For now, I just want you to know that the fluid content is maintained on the surface of the respiratory cells by this interplay movement of water, constant interplay of sodium, water, chloride, diffusing back and forth across the membrane. With water here, water out here, that is going to attract other water molecules in other places. For example, water molecules here are going to attract water molecules that are over here. And eventually those water molecules can pull on each other, causing this very large alveoli, where's that, to collapse. Okay, oops. All right. So this brings into mind another law, which looks at the relationship between surface tension and pressure. When we have a lot of surface tension, we have a greater amount of pressure, like it says here. Increased surface tension with water molecules attracted to one another increases alveolar pressure. Okay, so how does that bring us to the law of Laplace? Law of Laplace says that pressure, alveolar pressure, is equal to 2 times the surface tension divided by the radius. Okay. So now this doesn't necessarily apply to lungs. It can also apply to bubbles as well. And in fact, what this law says, if you've got 2 bubbles, if you've blown bubbles, the smaller bubble will actually have a higher pressure and it'll typically collapse much quicker than a large bubble. Okay, so why is that? Well, let's plug that into the equation. So there's a greater amount of pressure on the small bubble, and you can take a look at would be a greater amount of pressure in a small alveoli versus a large. But anyway, let's go back to this. Let's plug these numbers, plug some numbers into this equation. Say the larger bubble has a radius of 2, surface tension of 3. Plug that into this equation. That's going to give us 2 times 3 over 2. That's going to give us a pressure of 3. Smaller bubble has a smaller radius, same surface tension, but this translates into double the pressure. So if these 2 bubbles were, well, if they were connected to each other, right, this bubble would collapse into this one. But even if they're not connected to each other, this bubble will pop, will collapse. translate that into, so, and the same thing would be true with alveoli. Small alveoli will collapse, which we don't want any alveoli to collapse because that's going to affect our ability to breathe. So fortunately, we produce something called surfactant, which is like a detergent, and it's found in both, it's produced by those surfactant cells, those type 2 cells. It's found throughout the large alveoli as well as the small, but you can see right here, it's typically concentrated to a greater amount in the small alveoli. What does that mean? That basically means it has a much greater effect. So on what it has a greater effect is that it reduces surface tension to a greater degree in smaller alveoli. So if we take a look here, if here we have our alveoli with the radius of 2, that surfactant has reduced the surface tension from 3 to 2, which is great. So now we have a pressure of 2. We had 3 before, so that's good. But remember here we had 6. Because the surfactant is more highly concentrated in the small alveoli, it has a greater effect in the fact that it reduces the surface tension from 3 to 1. By doing that, we now have equal pressures in both large and small alveoli, which is going to prevent the collapse of those small alveoli. That helps to keep the lungs functional, helps to, by preventing them from collapsing. If they were to collapse, they wouldn't be able to reinflate. Sort of like if you've got 2 pieces of glass that get water between them, right? It's hard to pull them apart. Same thing here. All right. So looking at the big picture, law of Laplace says that alveolar pressure is directly proportional to surface tension, right? Greater the surface tension, greater the pressure. It's inversely related to radius. Alveolar pressure, greater pressure is found in smaller radii. And then I mentioned small alveoli typically will collapse, but they don't collapse because, as you guys know, we produce surfactant. And surfactant is a phospholipid complex with a protein produced by type 2 alveolar cells. By preventing the collapse, it prevents the collapse, of course, by decreasing surface tension. By reducing the surface tension, it increases the compliance, the ability to inflate. So that's a good thing. Oh, we're almost done. Okay. So let's talk a little bit more about surfactant and when it's produced and what happens if it's not. So surfactant production begins in late fetal life, typically after 34 or 35 weeks of gestation. If you think of a typical gestation of 40 weeks. Infants that are born before, like, are premature, before 34 weeks, typically have not produced any surfactant. They have difficulty breathing, and because they can't inflate their lungs. So this is why babies often have to be on a ventilator or an incubator if they're premature. Adults can also have this problem. For example, there's a condition called acute respiratory distress syndrome in adults, which can be caused by sepsis, a very serious systemic bacterial infection that can target the alveoli. And it does all these things, reduces compliance, affects surfactant production, has a very high mortality rate. And what happens is if you're not able to breathe very well, that's going to affect the function of the lungs and multiple organs can fail. So if a reduction in surfactant with this condition, reduction in surfactant also occurs with things like emphysema and pulmonary fibrosis. Fortunately, with technology now, certain companies have been able to manufacture artificial surfactant. And artificial surfactant can actually be delivered to people. This is an example of someone who had a condition this looks like sepsis. Can't really tell for sure. A lot of stuff is built up, just cloudy. This is after treatment with surfactant. It's definitely, the lungs are definitely clear. So that, fortunately, surfactant is available commercially now. The pathogenesis of acute respiratory distress syndrome is just like with a lot of our responses, or if our immune system gets a little bit out of whack, we want our immune system to work, right? But sometimes it becomes overactive at a particular tissue. You know, you've heard of autoimmune diseases. Well, this is not an autoimmune condition, but basically what happens is once you have this bacteria that enters into the system, this causes an inflammation, and that inflammation activates all these white blood cells, which start producing chemical mediators, which certainly can kill bacteria, but they also start a whole cascade of events, even clot formation. And if you take a look at that respiratory lining, remember I mentioned it needs to be very thin? Look at how thick it is. So this is believed, like some people that have had coronavirus, people with severe cases of that, this is also happening with coronavirus. All right. All right. So that takes us to the end of Respiratory I. Let me go back to here. All right. And I'm going to go ahead and upload this to YouTube, send this to you. Just remember, we've got Tuesday, we're going to cover more respiration. I'll be posting an updated lecture 2 and 3 somehow to sort of consolidate. And we'll have a review. I'll be sending out the study guide. So until then, have a great rest of the day, great weekend, and stay safe and healthy. Hasta luego.